Thank you for having me.
So, Dave is a software engineer. I am a software engineer as well. I'm a medical doctor too. But what Dave has done for medicine is Dave has added a new phenotype, and I'll let him explain what that is. A new phenotype to medical literature. I think that this particular new branch that Dave has introduced, we will be studying and working with this for a long time. So, Dave, thank you very much. You are a rock star for us in medicine. Please tell us a little bit about yourself and what is this Oreos and statins? What's going on here?
Yes, so I myself, as you mentioned, I'm a software engineer, I'm a platform architect. I actually had no interest in medicine or nutrition, anything of the sort, for quite some time. And in the process of working as I have for several decades in software engineering, I found that I was developing prediabetes. So my A1C was 6.1 for 2 years running, and I decided I wanted to do something about it. So I started to look at that time at what the ADA was recommending, what the Mayo Institute was recommending, and it didn't completely make sense to me given carbohydrates and the association with diabetes. And so I started looking into diabetic forms. Diabetic forms suggested possibly bringing down carbs, and low carb, high fat was pretty big at the time. Keto wasn't as big. This is in 2015. And after going on a low carb diet, I saw that my cholesterol went up substantially. And that got me extremely curious. It especially was curious because my dad and my sister, who started around the same time, did not see their cholesterol levels go up. And so I became obsessed with trying to figure out why. And as I told you off air, I then started looking into lipidology, the study of lipids and how cholesterol moves around in the body. And sure enough, it looks a lot like a network, which we can get into soon. But the model that I was working with and starting to build off of these experiments, I believe, allowed for us to make sense of this phenomenon. I'm proud to say that 9 years later, my good friend and collaborator, Nick Norwitz, managed to make a very entertaining and very interesting and thought-provoking experiment around it called the Oreo versus statin experiment. And I wish he were here. He's a very busy second-year med student, and he's got a very intensive month going on right now. But we want to keep the flame alive because as it dropped, the response has been tremendous. He's already done 12 interviews right before he got pulled into all of these rounds. The altmetric score, I'm sure you're probably familiar with that. We just saw that it's now crossed 1,750, which is just amazing. And that's been in just like a week and a half. The tweet where he put it out has now gone to over 1 million views. And the YouTube video that he's put out is over 500,000 views. So without question, it's drawn a lot of interest. So let me describe the experiment and we'll kind of work our way backwards because I think it'll make it a little bit easier. Basically, there are two interventions. The first intervention is he maintained his ketogenic diet, and he's very lean and has a very high level of LDL cholesterol since he'd gone on a ketogenic diet. Yeah, you can see him right here. And his cholesterol levels, his LDL cholesterol, the so-called bad cholesterol, would hover at around 400 to 500. So very, very high, close to homozygous FH levels. And this first intervention, what he would do is add Oreo cookies to his otherwise very low carb diet, his very low carb ketogenic diet, a sleeve of cookies a day. Again, this is hypercaloric, so it's on top of his existing diet. It's an overconsumption.
And he was
And
on keto at that time.
yes,
And then he switched to Oreos and the whole sleeve a day.
yes. Well, in addition, so again, technically still on his same diet, but with
Hmm.
the addition of the Oreo cookies. And he predicted, as well he should, per this lipid energy model, which we'll talk about in a moment, that it would dramatically lower his LDL cholesterol. Now, why would the addition of Oreo cookies lower his LDL cholesterol? Well, Very plainly stated, we believe that he's repleting the glycogen stores in the liver. That's the stored glucose levels in the liver. He's also increasing his insulin levels. This also increases leptin and a few other things that are part of the cascade. But the predominant central course of action that we're very interested in are the glycogen stores of the liver, because we believe that's upstream of all of these cascading downstream impacts that at the very end have a lot to do with your LDL cholesterol. And indeed, that's exactly what happened. The graphic you showed there, it showed that his LDL cholesterol had dropped 71%. He went from 384 on day 0 down to 111 16 days later. It's a massive drop. Now, that was the first intervention. And then he had about 2 months of a washout period before starting the second intervention. And note that those are days on the left side, those are 16 days, but they're weeks on the right side, for which he then took on rosuvastatin, 20 milligrams daily, which is a fairly high dose for that statin. And sure enough, at the nadir, the lowest point, that drop was just 32%. So it dropped from 421 to 284. So to put it plainly, the headline is that Oreo cookies dropped his LDL cholesterol twice as much as a statin. And this is a long span of time consuming the statin. And yes, sure enough, not only was that something that we were hypothesizing, but on top of that, it definitely garnered a lot of interest. There's now been a number of news stories that have carried it. Social media is exploding with it. And I'm so appreciative because Nick has just been doing a killer job of bringing a lot of interest and excitement around our research.
Very interesting. So to the audience, to the cool beans here, you know that we are very mechanism oriented. So I wanted to share, before we continue our discussion, I wanted to share with you Dave is here, Dave's Twitter. The links are in the description. This is Dave's YouTube, and I would recommend for you to subscribe to their YouTubes. This is Nick's YouTube. This is Dave's Instagram, and this is the study here. And why I recommend that you subscribe, you know that I very rarely ask you to subscribe to someone. When I started watching Nick's and Dave's videos about this LDL and this mechanism and the studies and the discussions that they were doing, these were amazing. I had to put my phone down to stop binging all the content that was present on these YouTube channels. The interest for me, for example, your analogy, the pizza box analogy with the VLDLs and the LDLs and the pizza in the box and the ApoB, that was amazing. And then Dave's discussions of the model were amazing as well. So number one, congratulations to all three of you, Cromwell, you, Nick. And then I have the next question. You became the trigger. Here is a study as well. In theory, this adds a new phenotype, a new group of people who are now discovered to have this particular set of numbers. Question which everybody asked me. So I've been discussing this with many people. Everybody said, will this high LDL in these individuals cause arterial and heart issues? Have you seen any studies around that so far?
Well, quite literally, we're the ones who are doing that study. There's one that's ongoing, that's nearing completion of its original scope, which will reach its completion of that scope this coming month. Actually, this month, it's February. I think we're actually a couple weeks away. And let me describe that real quick. So basically what we're doing is we're getting this phenotype you just mentioned. Let me actually unpack that first. When coming into this space, I would hear this term hyper responder for people who saw their cholesterol levels go high when going on a ketogenic diet. And I'd actually heard this from Dr. Thomas Dayspring and Peter Attia in articles that they'd written at the time, but there wasn't really that much information on this hyper responder phenomenon. Well, a couple years later, after working with a lot of these different labs that people were starting to send me after I'd started my blog Cholesterol Code, where I was sharing this, I said there really needs to be an expansion on this. It can't just be hyper responder, as in those people who see an increase in their LDL cholesterol, because actually the highest increases we see are those folks who tend to be leaner and more metabolically healthy, like myself and like Nick. And that also, by the way, differentiated me from my sister and my dad, which was also kind of relevant in looking at the physiology of this. So I called it lean mass hyperresponder. I published this article on my blog in July of 2017. And I really didn't genuinely know. Like I didn't know if it was just, I had only seen a couple dozen of these reports, but maybe there wasn't really that many more. Maybe I was wrong on how this applies back to the model. And that's not the case at all. It ended up becoming the most commented on blog post that I've ever put up. I have since started a Facebook group around it. It now has over 10,000 members to it. There's absolutely a phenotype that's very identifiable. And as described in that article, and as I'll describe to you now, it's not one, it's not just high LDL, it's three. It's high LDL alongside high HDL cholesterol, the so-called good cholesterol, and low triglycerides, which is a measure of fat in the blood. If you're actually at the site right now, put a little slash at the end, or actually can right there, lean mass hyperresponder. You can actually click on that link down there. It's right there in the menu, right underneath the header under cholesterol code. You can see lean mass hyperresponder. Yeah, I can show you that graphic right there for the lean mass hyperresponder. You can see those cut points right below. And again, it's amazing how much those first three items right above it, the low carb diet resulting in lower glycogen stores, the lower adipose stores, and the higher energy demands, those still really hold up to today. It kind of forms the basis of the model. And this phenotype, yeah, the question, as you just posed originally, was, okay, this is great, we're identifying it, but are they at a higher risk? And so that's why there's really two questions. The first question is the mechanistic. Are we right about the reason for the high LDL, particularly as it exhibits in the high HDL cholesterol and the low triglycerides? Okay, that's fine. But the second question is where is a little bit more pressing. This may still be right mechanistically, but are these folks at a higher risk? Well, you might be surprised to know this. Generally speaking, there just is no research around people who are metabolically healthy, but with very high levels of LDL, even before this phenotype was identified. It seems to me kind of obvious, frankly, that the moment you're determining that high LDL seems to be causal, you'd want to look at it absent other confounding factors. And the problem is that all versions of data that I've seen with extremely high LDL cholesterol before we've done the study we're doing now, they have some form of, they have a population that's either partially or exclusively focused for those who have some form of dysfunction in their lipid metabolism. Either they have metabolic syndrome or they have, for example, a familial hypercholesterolemia. But we don't specifically exclude those populations, instead focus on people who just have high LDL, but without some form of dysfunction. And lean mass hyperresponders have provided this new scientific opportunity because they don't just have a little bit of high LDL. They have very high LDL. Again, Nick is homozygous FH levels of LDL cholesterol. When you're up there at around 400, 500, modern medical science would insist that indeed he is at high risk. He has to be, because the lipid hypothesis would posit the higher you are and for the longer you are, the more heart disease you will have. And it doesn't matter if you're metabolically healthy. It doesn't matter if you're a non-smoker. All of those are risk factors, but it's mostly modulated by the high LDL. And so that's why we had to get this study going at Lundquist, because we need to find out for ourselves.
And I wanted to, while you're speaking and you're discussing this, I am just in awe. So I want to tell this to my audience once more. You are looking at a person and Nick and the remaining team who have added to medical literature a new phenotype. And I think this research and the outcomes of the research would continue for decades. So you are looking at him right now. Here he is. So thank you so much. I always get starstruck with individuals who have contributed to medical sciences and especially the mechanisms. So my question, medical science would insist that Nick, for example, should have the side effects of LDL. Has there been any calcium scoring done or anything done to kind of see what is the actual status of the body?
Yes. Well, of course, in the case of Nick specifically, he himself has had a CT angiogram. And to be fair, it was after 2.5 years at these elevated levels. But as of yet, there's not been any detectable levels of plaque that he's had with his CT angiography. That said, it's fair if critics would point to, well, wait a sec, this is a single case study. Case studies can be selective. That's why I wanted to do a prospective study. The Lundquist Institute study that we're doing, I wanted 100. I wanted 100 lean mass hyperresponders, or at least borderline lean mass hyperresponders, those who had this triad of high LDL, high HDL, and low triglycerides. And that will be much more informative because it's prospective, because we've described the phenotype, and rather than it being a case study where you can pull things from after the fact, you're actually getting it. You're actually going and getting it and then following the folks prospectively. And in that regard, I think it's going to provide us a lot more data specifically on plaque development. Now, I can tell you, I have already presented somewhat, and now Dr. Budoff has presented on what's actually going to go to publication, which is this match analysis. And of our cohort, we do have some folks who do have plaque, but we kind of expected that given they were at age 55. So there was going to be some amount. You can get any group of people that's going to be average age 55 and whatever diet they're on, whatever their circumstances, they're going to be more likely to have plaque than not. But that said, the majority of ours have no plaque. The majority of our cohort actually has a total plaque score of 0 out of a total of 45. And the remaining, except for one, have a total plaque score in the single digits. And only one, the very last one, has one in the double digits, and that's just a 12, again, out of a total score of 45. So I will concede it's been looking pretty interesting so far in that it doesn't appear to be a high-risk population, but it is still early days, and we still have more research to go.
So could this be, we say in medical sciences, before your phenotype's introduction, that LDL is circulating, and then we can have the cholesterols going into the blood vessel walls, and then over there the macrophages would pick it up, and the foam cells would form, and inflammation would occur, and atherosclerosis would occur. In the case of these individuals who have this high level of LDL, is it possible that the cholesterol and the other ingredients, triglycerides, are actually basically picked up by the cells? And so even if the numbers are higher, they are nourishing the cells instead of getting into the blood vessels? Or are they empty? What is your theory or hypothesis, you and teams?
Well, I can't say that I speak for all of my co-authors, but I can tell you, for me, I'm very interested in how the entire class of ApoB lipoproteins are utilized by the body. And I know it's often talked about as though, once general lipid metabolism is used, LDL is almost like the leftover garbage, if you will, and it's about trying to clear it out as fast as possible. And I don't want to be dismissive of it. It's very possible, but I do. There are a lot of things in literature that show it's potentially a component of the immune response. I think that's probably the case, particularly given that you find that they carry alpha tocopherol, which of course is vitamin E, that actually is an antioxidant. It actually protrudes their monolayer, the helix protrudes. So if it comes into contact with reactive oxygen species, for example, it'll turn both it and the ROS into a non-reactive product. LDL particles bind to pathogens. And one of the more interesting things that's now been more acknowledged in the last few years is that sites of inflammation, you were talking about endothelial cells, they actually upregulate the binding to LDL particles for a process known as transcytosis, trans being transportation, cytosis being a process of the cell. At sites of inflammation for unmodified LDL particles through a receptor known as SRB1, that's extremely relevant and interesting to me because, for one, that same process happens even when there's not inflammation at a lower amount. Endothelial cells will invaginate these LDL particles and transcytose them to the other side. But if it's at a site of inflammation that it happens at a higher rate, I think that's very relevant when you're looking at the process of atherosclerosis, given we already know it's an inflammatory disease. The challenge is that there's a bit of an argument as to which is happening first. We know if we damage the endothelium period, you'll have resulting atherosclerosis. So step 1 is the damage, step 2 is the inflammatory response, and step 3 is the development of these plaques. We know this from animal models. We do it all the time, right? We don't know if the concentration gradient of LDL particles in an otherwise functional lipid metabolism will induce the development of plaque independently. It's, of course, the prevailing lipid hypothesis. And again, I don't want to be dismissive of it. It's just I myself have been waiting for this exact moment where we would have people who seem to have higher cholesterol due to metabolism because they literally are metabolizing more fat and therefore systemically needing to traffic it more per the lipid energy model we're talking about. Because if it's true, if a concentration gradient is, all that's needed, then for sure, the lean mass hyperresponders should demonstrate a higher amount of atherosclerosis in spite of having a lower level of inflammation. But I don't know. We'll have to find out.
And we'll have to continue to research and follow these individuals. This is such an interesting area. Just to connect it a little more closer to myself, my older brother is leaner. He's always been lean, and he's always had high LDL. And doctor has always said this seems to be a genetic issue with your genetics, and that is why it may be high, and has always insisted that you take statins. And this has been happening even when he was 31 years, that he's in that. So I actually sent your research to him, and I said, you should actually go figure this out, that maybe you are this phenotype as well. So I have a question for you. The pizza box model, if you can enlighten us a little about that. I really loved it. I was actually thinking of asking you that, could I please use that with proper attribution to you as well? So tell us a little about that model.
Well, part of the challenge when people are needing to think about this process is they need to think of lipoproteins, particularly LDL, as ultimately carriers, that it's a system of shipping, right? So I have kind of another way of putting this since I have a little more time to unpack it. The ApoB lineage of lipoproteins, you can kind of think of them as sort of a protein. ApoB is kind of a protein that has a complex of things attached to it. I know we tend to think of ApoB as being on the lipoprotein, but I would argue ApoB is the only thing that's truly fixed and immutable. Everything attached to the ApoB is cargo, right? So in a sense, the ApoB is like the pizza box, and everything that's inside is like the contents of the pizza box. Well, I would argue that an ApoB's job is to start large and to get small. How does it get small? Well, because it's dropping off its cargo. It's making deliveries. So it'll either leave the gut, your small intestine, or it'll start out in the liver, which is predominantly with lipids from storage. And the one that leaves the liver is the one we're focused on a lot, which is a VLDL. And a VLDL, by its nature, loaded up with lipids. It's out there. It's ready to drop it off. Now, under good conditions, it will succeed at dropping them off and dropping them off quickly. The cells all around your body are like, good, good. I've been needing lipids, right? So they're kind of like hungry customers anxious for their pizzas. Well, if you're succeeding at dropping off the lipids from those ApoB, then it's kind of like people eating the pizza out of the boxes. You have something left over. You have the container, but it's an empty container, right? Just like you have the empty pizza boxes. Now, if you need to deliver more lipids, you're going to have to send out more ApoB lipoproteins to keep resupplying, right? And if that's the case, then it makes sense that you'd see more ApoB. By the same token, if you've got, a house full of hungry kids for a birthday party, you barely get those pizza boxes in the door, and they're just eating all the pizza out of the pizza boxes. So you have a whole bunch of leftover empty pizza boxes, right? So that's kind of the gist. Now, what's really interesting is we can just very quickly swap out what the pizza is on the inside with triglycerides. Triglycerides are the stored form of fat. It's got, if I can speak geeky for a second, it's got a lot of ATP potential. I think a ketone body is something like 22 potential ATP yield. I think a glucose molecule is going to be anywhere between 32 and 38 ATP yield. A triglyceride, fun fact to know and tell, it can vary based on the length and the type, but it's probably around 460 ATP yield, right? And
wow, I didn't know that.
Another fun fact to know and tell, guess how many molecules would be on a typical VLDL1, a mature as it's leaving VLD1? 45,000. So 45,000 times 460 ATP yield. These things are tankers of fat-based energy. They're gigantic, right? And if you're like myself or Nick, what you have are these little food banks all around called subcutaneous fat cells. And the more you have lean mass and the less you have fat mass, the more you have to recirculate. It's a redistribution scheme. So these carriers, they're more active. There needs to be more and more rapid shipping. How do we know if it's succeeding? We know it's succeeding because if you get a snapshot at any moment in time, you'll find the triglycerides are low. They're not low because there's less getting shipped out. They're low because in spite of how many are getting shipped out, they're getting turned over very quickly. And how do we know that they're getting turned over very quickly? That's the best part, is the third component, is HDL cholesterol. HDL cholesterol goes up. Do you want to know why HDL goes up per the lipid energy model, as we would posit? It's because they are the part of the shipping that is in the reverse of ApoB. ApoA1 is the major protein to HDL. And HDL is a fleet of boats that are nearby that their job is to start small and to get big. They start out very small. They almost look like a cookie if you ever see a nascent HDL. And this process of turnover, it actually releases a lot of components that HDL particles pick up. Another fun fact to know and tell, you actually have many more HDL particles than you do ApoB particles at any given moment. But the cholesterol esters, when you see HDL cholesterol, what they're talking about are the cholesterol found inside HDL. And that's why when triglycerides go down, typically you see HDL go up. Why? Because it's turnover. It's successful turnover of those VLDL releasing those components into HDL. Now, I know that's going to be unusual to a lot of people in your audience, because what we learned growing up was that HDL cholesterol is probably because they're picking it up from the tissue. And that does occur. But much of HDL cholesterol that you're going to find right now in your bloodstream is due to the turnover of VLDL, especially into LDL and other ApoB lipoproteins, that turnover process. And that's what creates the triad. That's what creates the high LDL, high HDL, low triglycerides. And that's why I think this phenotype is so important, because only in the circumstance where people would choose to become fat adapted would these things line up as they have across all these ethnicities, all these ages. Um, and it's really kind of exciting, honestly. The engineer in me is very tantalized by it.
Excellent. So I really love this. I'm going to ask you one more question. And there are cool beans over here, and they are totally impressed by this work. Some of them are impressed by your dimples as well. So a lot is happening here. So I have a question for you. This is a normal human reaction that when something new comes in, some of us would quickly adapt it and say, yeah, this is awesome, and try to understand that more. Some of us would say, no, not good. What are you talking about? You're not in the right direction. How is this research and this new phenotype and this new model that you are proposing, how is this received? Are there challenges to this model? Are there more acceptance? How do you see it?
Well, yes. First and foremost, the most common thing that was thrown in my direction early on was it's really just saturated fat, because it's been assumed that those who see this response, including the early days of where I was reading it from doctors Peter Attia and Thomas Dayspring, was that was the intervention du jour, was to bring down the saturated fat. However, as Nick has shown, because his diet is actually more Mediterranean style and most of his fat comes from extra virgin olive oil, it really doesn't seem to line up much at all with saturated fat. But on top of that, we recently published a meta-analysis of 41 randomized controlled trials where at least one arm had an intervention of low carb. And in that meta-analysis, we did show that saturated fat did not have nearly the same effect as BMI, which lines up with the model. The leaner you are, the more likely you see this pronounced response. And this is somewhat evident by the fact that currently there already are anecdotally not a lot of people who start out, say, obese and come to a place of being overweight. They haven't yet gotten lean, but they themselves will often say that either their LDL hasn't changed or that it's even gone down, especially if they started in a more metabolically challenged state. So we would see we would see folks who went from a mixed diet to one that had a lot of saturated fat have the kind of responses that Nick and I have had. But that's not common at all. If you've been inside the Facebook group for lean mass hyperresponders, I had people send random photos for us to just use for the main banner, and I just grabbed them at random. But if you didn't know that this was a group that came together around lipid profiles, you would assume it was some kind of fitness group because everybody looks like they're more athletic, they're leaner, they're more focused on health. But that's also what is kind of exciting, is it became an almost hat trick that if people sent me their lipids in the early days and they sent me something that looked like a lean mass hyperresponder, I would just straight say that to them. I would say, so you're probably lean and maybe even a little athletic, right? Given this profile. And they'd go, how would you know that? Because your lipids are, the profile is very suggestive of what's going on with you internally, metabolically. And that's part of what makes it exciting.
Excellent. This is amazing. So I think we'll have to request you, Nick, your other team members to join us more and more. And I would actually, once again, just a quick introduction to your presence on the social media. So this is the study, Cool Beans, and I have the link to this study in the description of this video. This is Dave's Twitter. This is his blog area. This is Dave's YouTube channel. This is Nick's YouTube channel. This is Dave's Instagram. So you are fully aware of where these folks are. I am, of course, as you can tell, starstruck by their contribution to the science and their work on it that is ongoing. So I'm going to just go through some of the comments as well and see if there are any questions. Of course, you brought it onto yourself because you talked about Oreo cookies. So if you have fun questions, that is all. So how about Oreos covered eggs? Will that help?
I mean, I suppose it. Here's one thing I've learned about Nick. He's not only willing to do a lot of different experiments, but he's willing to try a lot of different foods that I'm not sure if I would have the same palate for. But yeah, I wouldn't be surprised if we could talk him into trying Oreo covered eggs. I myself have never really liked Oreos that much, but it's always possible I might try to replicate an experiment of his just so that we can at least have an N of 2. So I suppose that's possible.
Understood. So Lil Fella says, how about Chips Ahoy Chunky?
Oh, now see, now you're talking my language. That's an intervention I could get behind. But yes, per the lipid energy model, we would anticipate that if it has a decent amount of carbohydrates, that's exactly what we'd be going for, is to try to replete the liver stores. It's worth emphasizing the reason why Oreo cookies were chosen is for the same reason that I myself have done a version of this, which is the white bread experiment. It's to kind of intentionally pick something that people wouldn't consider healthy, because a lot of times if somebody leaves a low carb diet and they're going on, say, like a plant-based diet, a lot of times the claim is, oh, well, that's because, certain phytonutrients or there's more vitamin C or something like that. And part of the reason for this kind of an experiment like Nick is doing is because it's saying, no, what we're pointing to is the physiology, and we're intentionally choosing something that's, not strongly considered to be healthy on purpose, and maybe a little bit
Understood.
clickbaity, right?
Understood. As he said that this was a head turner, and this definitely is. And I want to put this disclaimer out that we are not talking about over here that Oreos are better than statins or Oreos are better anyways having that sleeve. I think it depends upon the person's physiology, their phenotype, their body habitus, what is their state, and then what is their metabolic model, and in which a carbohydrate rich diet could make an unexpected turn towards the numbers, the cholesterol numbers. So that is what is the discussion. That is the new discovery. That is what is exciting me as well. So here, there's a question I cannot pronounce. Socrates, probably. For how long were these individuals on a ketogenic diet before the study?
So in the case of Nick, I believe he's been on a ketogenic diet 4.5 years, possibly 5. I'm not sure. Myself, per what I mentioned earlier, since 2015. So I've been about 9 years in at this point.
Understood.
Yeah, and for what it's worth, we would posit that it's almost as fast as you become a lean mass hyperresponder. We would anticipate a comparable response if this happened. But I am glad you brought that up, and it is worth reemphasizing. This was meant to be a demonstration. This experiment is not actually meant to be taken seriously as in, in any way a recommendation. This is a don't try this at home. Nick did this so we don't have to.
Understood. Thank you. Makes sense. And John Snyder says, how about chocolate goodness instead of Oreos?
Oh yeah, again, I'm on board with that as well. I like virtually all versions of something chocolate. So I would agree. That would be goodness as well.
So I have a question. Is this a genetic model, or anybody who's lean? So imagine if I'm not lean, and if I become lean and then I am on keto, would I end up with this metabolic model as well, or this is just a genetic predisposition?
That's probably one of the other bigger pushbacks is, now that there's more acknowledgement, it's not just saturated fat and that there really does seem to be some physiology. There's some folks who say, well, this isn't happening to everybody, and therefore we think this is some unusual genetic predisposition. Maybe hyperabsorption is probably one of the more common ones that are said. But we would propose that no, if you gave us let's say a gym down the street, that we could just choose a bunch of people that would meet the phenotype we think externally. So they're like Nick, they're lean, they're very aerobic, they're very active, that if we could just choose them And then we control their diet. We put them on the diet that we want. We control for a few other confounders. For example, I wouldn't want them to do intensive weightlifting that can lower LDL. But if I can control the diet, I believe the vast majority would likely see the change to this phenotype. And I say the vast majority, not 100%, because I think that there are still other factors. So if I had to bet, I would bet that, yes, if you gave me total control of your diet, I could take you to Dave Island and, dictate, just like if you were an animal model. I think I could. I think I could turn, let me put it this way. I think it's more probable than not that just about anybody could become a lean mass hyperresponder.
I suspect so as well, because if we look at your metabolic model, if that is just with the genetic predisposition, and again, conceding that that is a hypothetical model and you'll continue to prove it or disprove it, if it was genetic, then you would not see that in only lean folks. You would see that in many other folks as well. But having a specific body habitus and the body metabolic state, and then using carbs to show how the LDL goes down, to me, it seems like more of a body habitus than the genetic, but this is just my thought. So let's see if there is any other extra dark chocolate as well. So with this, I think that we have a lot of fun with this idea of Oreos reducing LDL more than cholesterol in this specific phenotype. But at the same time, thank you very much to you, to Nick, to William Cromwell for doing these researches. Thank you for you to kind of sticking to it to say, what the heck is going on? I want to understand more. And then ending up contributing to the medical literature. Any thoughts? What are your future plans with this research? Where are you going with this?
Oh, well, definitely the most important thing to me, especially in the near future, is for us to finally get this longitudinal data back on risk. I know I kind of peppered through our conversation this Lundquist study. It's worth emphasizing that it's longitudinal, which is to say everybody's getting a total of 2 scans, one at day 0 and another around 365 days later. And the last of our participants to get their second scan, that's going to happen this month, and it'll happen within a couple weeks. That's going to be very important data because, again, the present medical version of the lipid hypothesis is we should see a rapid progression of plaque at a population level for these folks who are in our study. And if we don't, then that's going to be a major, major first step. But on top of that, we're also raising money for a companion study, a study I'm going to call the Triad study that we have a conference for. That's actually next month. It's in March 15th and 16th here in Las Vegas. And it's a fundraising conference. Literally, we're taking that money and we're turning it right over to this Triad study, this companion study. And if we get 2 studies by that point, my hope is just that we get more interest above and beyond us to study these folks, because I really do think it's more than risk. I think if they prove to be low risk, on top of everything else we're speculating mechanistically, they might teach us a lot about lipid physiology, and that's exciting. If we're right, we'll see.
Absolutely. Not only it would teach us about this new phenotype, it would actually shed a lot of light at the current inflammatory process and the understanding of that and how that may be happening and what is the role of LDL in there or not. So that all is very exciting. Thank you very much for doing this discussion with us. Thank you for being here. Thank you for doing this work. I hope that as you come up with more research and more data, we could continue to invite you to come and share that with us as well.
That would be fantastic. Thank you for having me on.
Thank you very much. So Cool Beans, thank you very much. As you just heard, here is Dave with a new contribution to medical literature and medical sciences. And these are the rock stars that we love to talk. They increase the ambience of our neighborhood as well. Thank you very much. And I would see you tomorrow. Bye for now.